From the last chapter, we have looked at cellular respiration, where we are able to harvest energy in the form of ATP along with carbon dioxide and H2O from one molecule of glucose with the presence of oxygen by series of steps of reactions. In Chapter 6 for photosynthesis, we will look at how glucose is produced by carbon fixation and how energy from ATP is stored in the glucose which will be consumed by the consumer. So, photosynthesis can be defined as the chemical process by which green plants and other phototrophs synthesize organic compounds such as carbohydrates from carbon dioxide and H2O in the presence of sunlight. In this photosynthesis process, we will convert light energy to chemical energy. We have the light energy from the sun, and this energy will be converted into chemical energy in the form of ATP and into glucose. Photosynthesis occurs in the chloroplast. There are two reactions in photosynthesis, which are light-dependent reaction and light-independent reaction. Light-dependent reaction, based on the name, this reaction need light energy, and this reaction occur in the thylakoid membrane. While for the light-independent reaction means that this reaction do not need light energy, and this reaction occur in the stroma of chloroplast. Both of these reactions are needed to achieve the goal of producing glucose with a high energy level, and that is the process of photosynthesis. So in this video, I will look at light-dependent reaction first, where I will explain the cyclic and non-cyclic photophosphorylation. In general, light-dependent reaction uses light energy, mainly from the sun, to produce ATP and NADPH. Similar to NADH in cellular respiration, NADPH is also an electron carrier. Light-dependent reaction occur in the thylakoid membrane, and it involves photosystem. Photosystem is a protein complex that consists of two parts, light harvesting complex and reaction center. The purple one is the light harvesting complex and the blue one is the reaction center complex. A light harvesting complex consists of pigment molecules such as chlorophyll B and carotenoid. This pigment will harvest the light energy. The reaction center complex consists of a pair of chlorophyll A molecules, means that we have two chlorophyll A molecule that is paired together and a primary electron acceptor. This is where the electron transfer will occur. And we have two types of photosystem in the light-dependent reaction, photosystem 1 and photosystem 2. These two photosystems are differentiated based on the ability of chlorophyll A to absorb either 700 nanometer wavelength of light for photosystem 1 and 680 nanometer wavelength of light for photosystem 2. There are two types of reactions in the light-dependent reaction. The first one is non-cyclic reaction. This reaction is linear, where we have the first electron acceptor and the final electron acceptor, which is NADP+. The second reaction is cyclic reaction. This reaction involves the electron continuously used without any last electron acceptor. Let's look at non-cyclic reaction first. As you go through this video, try to fill in your bio treasure. Okay? A non-cyclic reaction involves three processes, which are photoactivation, photolysis, and photophosphorylation. Why photo at the front of each of the processes name? It basically to inform that these processes occur with the help from light energy. And this reaction involve photosystem 1 and photosystem 2. The first process is photoactivation. The antenna pigment in the light harvesting complex in photosystem 2, this is photosystem 2, absorb the light energy or photon from the sun and pass the energy from one antenna pigment to another antenna pigment until the light energy reach the reaction center, which is the P680. Why P680? Because this is photosystem 2. When the P680 reaction center receives the light energy, it undergo photoactivation and release 2 electrons to the primary electron acceptor. Here, P680 oxidize as it donate electron and become P680 plus. Then, photolysis of water occur to produce oxygen that will be released to the environment, hydrogen ion that will be released to the thylakoid space, and 2 electrons. These 2 electrons will be donated back to the P680 plus to reduce it back into P680. The electron from the reaction center of photosystem 2 go through ETC before they reach photosystem 1. As the electrons are passed through the ETC, from plastoquinone, cytochrome complex, and plastocyanine will release exogenic energy, and this energy will be used to produce ATP. At the same time, photon is absorbed by antenna pigment in photosystem 1. The photon is transferred from one antenna pigment to another antenna pigment until it reaches the chlorophyll A in photosystem 1, in the reaction center of photosystem 1, that is P700. P700 undergo photoactivation and release the photoexcited electrons to the primary electron acceptor. P700 is oxidized to P700 plus because it donates its electron. Electron from the P680 of photosystem 2 that undergo ETC, from PC, the electron from PC will replace the lost excited electron from P700. So, P700 plus will be reduced to become back into P700. Primary electron acceptor that receive electrons from P700 will transfer the electron to another ETC in the thylakoid membrane, that is the ferredoxin. Ferredoxin will transfer electron to NADP+ with the help of NADP+ reductase. NADP+ will be oxidized as it receives electron and transform into NADPH. NADPH will become our electron carrier. Those are the 8 steps in the non-cyclic reaction. And from this reaction, we will have ATP, we will produce ATP, NADPH plus H plus, and oxygen, which is the byproduct. Now let's look at cyclic reaction, and this reaction involves photosystem 1 only, and it will only produce ATP. Same as in the non-cyclic reaction, photon is absorbed by the antenna pigment molecule and it is transferred until the photon reach chlorophyll A P700 in the reaction center. P700 undergo photoactivation, release the photoexcited electrons to the primary electron acceptor. P700, as it donate its electron, it becomes P700 plus. Primary electron acceptor will then pass the electron to ferredoxin. Instead of NADP+, ferredoxin will transfer the electron to cytochrome complex. Cytochrome complex then transfer the electron to PC. As the cytochrome complex pass the electron to PC, it will release exergonic energy, and this energy will be used to produce ATP. PC will then transfer the electron back to the P700, thus continue the cycle until the cell have enough ATP. Cyclic reaction only produce ATP, and usually plant cell switch from non-cyclic to cyclic reaction, usually when the cell is low in ATP, so that they can produce more ATP from this reaction. In conclusion, the ATP and NADPH produced from the light-dependent reaction will be used in the light-independent reaction, or also called as Calvin cycle. Calvin cycle is a carbon fixation process where we will convert carbon from carbon dioxide into organic compound, that is glucose.